Welcome back to the Soil Health Dimension series. My name is Hunter Francis at Cal Poly State University in San Luis Obispo, California. This series is part of a USDA SARE-funded project and has been made possible by numerous other funders who are listed on the homepage of this online series. Today we're going to be talking about the assessment of soil health. Now that we know that the soil in large part is a biological system, it should not be a surprise that assessment is difficult. It's literally a moving part. So there are many ways that one can approach it in trying to assess health status. It is and always has been important to test agricultural soils for a number of reasons. Ultimately, our goal is to get as much investment into the plant as possible. Ideally, well over half of what is spent on inputs should make its way into crops for sale. But in order to avoid waste, it's important to know what your starting point is, and this can only be done through testing. Good management involves good measurement of what the variables are within the system. Since we're talking largely about fertility here, a big focus for a long time has understandably been on the nutrient status of soils. What's been pointed out in this series is that is just part of the picture, and the soil biology has a big role to play in nutrient delivery. Many of the new assessment methods and management practices that are now being promoted throughout agriculture take that into consideration. Some of the methods, both old and new, can be conducted in the field fairly easily and rapidly, but many of them still require the assistance of an accredited laboratory. Assessment not only helps at the start of a season or an agricultural venture, but testing at crucial points throughout the season can also help monitor the status and progress of a crop. It can also help in future decision making, especially when good record keeping is combined with testing. Keeping track of past successes and failures helps one to identify trends and what's working and what's not. In the end, this precision can have a big effect on an operation's bottom line. As always, organic matter, or OM, is the star of the show, and that is sometimes put in different categories because it comes from biology but can be tested chemically. It's sometimes what is called a biogeochemical indicator. Some main categories of newer biological indicators include biomass, diversity, and activity, which we will define and discuss, potential contributions of bioavailable carbon and nitrogen, respiration, which is also a form of activity, and new genetic testing techniques. This slide was shown in section 1 of this series and lists some new soil health indicators that are being considered for recommendation by the NRCS. The procedures for testing each are currently being vetted by a national team of leading scientists, and it's possible that some of the recommendations may be refined. However, these parameters are being reflected in many different frameworks for soil health assessment, and it's likely that managers will be incorporating them more and more into decision making. Again, the exact list of tests used on any one operation will vary according to circumstance and preference, but these are some of the main parameters that are being worked with. We've talked quite a bit about the physical and chemical, so this section of the series will focus more on the biological tests, many of which are new or just starting to be applied to agriculture. We will nonetheless review a few of the key points about some of the physiochemical parameters. One of the most basic soil health indicators, whose importance has been understood for a long time, is bulk density. Density is simply the mass of a substance divided by its volume, and in the case of soil is usually referred to in grams per cubic centimeter. As any given soil achieves more tilth, it lightens up. There's much less mass per given volume because there's more pore space. Aggregation, which is the opposite of compaction, helps to achieve this. Bulk density is still used as a measure for tilth and structure, especially over time when trying to assess the effects of new management practices. This is a picture of two people taking soil cores, normally done with a metal cylinder of a given volume, that's gently pounded into the earth and then dried and weighed. From that, bulk density is calculated. Another relatively simple technique is the use of a penetrometer to assess the relative ease with which the soil can be penetrated. This is also a measure of tilth and how compacted a soil may be. The graph on the bottom of this page gives an estimation of the ability for roots to penetrate a soil at a certain penetrometer readings. Normally, of course, it will take less pressure to push the probe into the soil when it is not overly compacted. Oftentimes, it will take different pressures to penetrate the soil at different depths, and this can help identify hard pans beneath the soil or surface compaction, and can help with decisions about tillage. There's a link to a short video on how to use a penetrometer that was produced by the University of Wisconsin, listed on our webpage for this chapter. There's also a resource listed from Cornell University where one can find guidelines on how to sample soils. This also includes a how-to video. These recommendations are being used by hundreds of participants taking part in their National Soil Health Assessment Project. Good soil sampling technique is important in getting good results from laboratories or even for one's own use. Sampling technique in part depends upon what one is looking for, but in general, unless one is trying to troubleshoot a particular problem in a particular area, it's good to get a representative sample from the entire block that one is working with. If the soils within an acre block are highly variable, as many as 20 or more samples might be taken and mixed together to try to achieve an average. A field that is more homogeneous or consistent may require half that many samples. Different labs will have different recommendations. The depth of the soil taken also may vary upon crop. Samples for shallow-rooted crops normally are taken within the first 6 inches of soil, where deeper-rooted crops may go down to a foot or more. It's customary to remove the top 2 inches of a sample before mixing it, in any case, to avoid any anomalies occurring at the surface. This, of course, involves removing any litter at the top of the soil even before the soil sample is taken. Depicted here are a few main instruments used in soil sampling: soil probes, augers, or shovels. Lysimeters have been used for a long time, often to measure water potential in a soil. They are a form of in situ chamber, in other words, one that resides in place in the soil, and allows the soil solution to penetrate it, where it can be collected and measured. Soil solution thus gathered is increasingly being used for nutrient analysis, and in the future will be able to be monitored remotely in real time. Above the ground, soil nitrate quick tests are increasingly being used with good success. They are easy to use and relatively inexpensive, and a good way for growers to supplement lab analyses to do quick reads of residual nitrate currently available in the soil system. The test strips used are not unlike litmus paper for pH, or even strips that people use to test chlorine in pools. A soil solution is made with about 3 parts calcium chloride solution and 1 part soil that's agitated and dispersed, and then exposed to the tip of a test strip. Immediately after shaking off excess water, it's important to compare the color on the strip with the color scale provided within 60 seconds or so, as the color can change over time. It's also important to do the test soon after mixing the soil into the solution, because soil microbial activity can release additional nitrogen if it is left to incubate too long, potentially throwing off the results of the test. Results are given in parts per million. Since, as we've learned, a foot of soil across an acre is approximately 4 million pounds, 1 part per million nitrate would equate to 4 pounds of nitrogen per acre. In other words, a part per million test would theoretically be multiplied by 4 to get pounds per acre. However, there are correction factors that are used to adjust for differences in soil type and moisture content, so one should follow the recommendation of the quick test provider. We've included a local resource here as an example recommendation that explains how this test is done. What's hopefully quite clear by now is that given the activity of the biology in the soil, residual nitrate that exists in the soil solution at any one time is only part of the nitrogen contribution to plants as they grow. Therefore, a lot of work has been done in the scientific community to try to get a handle on how to predict additional nutrient that will be added to the system as nutrients are mineralized and released by the soil biota. In the case of nitrogen, this has resulted in the concept of potentially mineralizable nitrogen, or PMN. This test is usually done in the laboratory because it involves precise monitoring and incubation times of a week or more, and tracks how quickly nitrogen is being released by the soil biology. Results then give an indication of how much nitrogen exists within soil organic matter that can readily be converted into a plant-available inorganic form. As one might expect, soil with low amounts of organic material, or soils that have been poorly managed, will have lower reserves of organic matter that's readily available to be mineralized. How nitrogen is mineralized is often affected by moisture conditions. In aerobic soils that contain sufficient oxygen, nitrate is produced. In anaerobic soils, such as wetlands for rice, mineralized nitrogen is in the form of ammonium. Also, not surprisingly, science is confirming that higher levels of potentially mineralizable nitrogen correspond to the presence of beneficial bacteria and even higher organisms that are helpful, such as beneficial nematodes. In terms of fertility, there are, of course, other ways of assessing nutrient status once a crop is underway. Tissue and sap tests can be useful in assessing how well nutrients are being assimilated by a crop and how they are being translocated within the plant as it grows. Tissue tests can be useful in identifying real-time deficiencies. New technologies on the horizon are also making their way into agriculture with some success and will undoubtedly be used to an increasing degree as their precision increases. Remote sensing by satellites, drones, and implements mounted on machinery often use optics or spectral analysis to help in a variety of ways. Differences in light readings can be used to identify crop stress, including water stress, and even quantify nutrients present in crop tissue. Combined with other tests, visual analysis, and site history, these tests are becoming better correlated to actual conditions and thus are improving in their efficacy. In terms of quantifying actual microbes in the soil, visual inspection and the counting of organisms began with the invention of the microscope in the 1600s. There are now much more sophisticated ways to count organisms that can't be seen by the naked eye, such as by suspending them in a solution and running them through a counting machine. This is a technique called flow cytometry. Another traditional way of understanding who was down there was through plating them and growing them out in a laboratory. Tissue culture enables one to isolate and estimate the relative density of a particular organism with a sample, and also, as in this picture here, enables one to identify organisms that would grow on certain substrates or food sources. Part of the problem with tissue culture, however, is that certain microbes do not grow well in the laboratory, so it is not really possible to do justice to the whole range of organisms in the microbiome. Getting a handle on larger organisms is, of course, easier. The return of earthworms into a soil that has been degraded is an excellent indicator of improved soil health. Nematodes, which are smaller and barely visible to the naked eye, are also being used fairly frequently in some soil health assessments. They are extremely prevalent in soil systems, mostly beneficial, yet exceedingly diverse given their many hundreds of species. Use of nematodes as an indicator is based on the fact that they can be classed or grouped according to their mouth parts. This helps to determine whether or not they are beneficial and whether they are contributing to soil health. When plant parasites predominate, for instance, that is an indication of an imbalance that can have negative consequences for agriculture. In Chapter 2, we discussed the huge diversity and abundance of microorganisms in the soil and the fact that the vast majority of them, as much as 95% of them, have not been identified. Enumerating individual species of the smaller microbes would be daunting. Therefore, a common way of grouping them together to quantify them as a whole is by referring to them and studying them as biomass, which is just the sum total of the weight of the organisms in the soil. Biomass is largely composed of bacteria and fungi because they are normally most prevalent, even in terms of weight, and, as was mentioned previously, make up about 5% of the carbon in soil organic matter. Most of that organic matter is dead, more stable carbon that has been degraded. About half of the biomass is in the top 6 inches of soil, and one of the reasons it has become the useful indicator is that it can help in the assessment of fluctuations in soil carbon over short periods of time. Older, more degraded and stable portions of the organic profile tend to shift more slowly. Therefore, biomass is more apt to be reflective of effects resulting from changes in management practices. If a beneficial practice, such as cover cropping, is actually working and having a positive effect, one would likely see increases in biomass before one saw significant increases in total organic matter. That might take longer to build. The main way of assessing biomass is through chloroform fumigation extraction, which essentially lyses and extracts the guts out of microbes so that they can be assessed for total nitrogen or carbon. The resulting quantification of how much biotic substance is present is sometimes called abundance. Sometimes in talking about biomass, one might refer to the potential versus the attainable versus the actual biomass. The potential biomass refers to the most biomass that one might hope to attain given a certain soil type and where one is in the soil profile. Clay soils with more pore space would tend to increase the potential, whereas increasing depth decreases the amount of biomass that one might expect to find. The attainable biomass refers to what might be a realistic given environmental conditions, namely moisture and temperature. Where there's more moisture, microbial communities are more prevalent and have the ability to build up when temperatures are cooler. Finally, the actual biomass that one has in a soil is what we have the most effect on through management. Good soil building practices that keep the soil covered as much as possible and that build organic matter will tend to build biomass. Numerous parameters or variables affecting biomass could be considered and studied. In this case, on the lower right, the effect of CEC on biomass was considered. Not surprisingly, higher cation exchange capacity tended towards higher biomass, and this is related most likely to finer soil textures and greater organic matter content in the soil, both of which help to increase CEC. Microbial diversity refers to who is there as opposed to how much biomass exists. Higher diversity can be an indicator of soil health because with more diversity there is more likelihood that a wide range of soil functions are being performed and perhaps with more robustness. Redundancy of tasks is actually a good thing, and can be a strength in the soil system, because if there are multiple groups performing the same task, then if one of them gets knocked out for some reason, there are other organisms who can get the function done instead. On the lower right are some indicators of how diversity can be studied, such as by simple diversity, how evenly distributed the populations are, how rich they are, meaning that there is good representation within each group and not just a shallow veneer of many different species, or whether, on the other hand, one group of organism dominates. This study looked at how temperature affected these different aspects of diversity, and with a slightly higher temperature, found that there was an increase in diversity and evenness with less dominance by relatively fewer species. The most common way to assess diversity is by studying the cell membrane of the organisms involved, and this is because the chemistry of the phospholipids that make up a cell membrane are characteristic for each species. This phospholipid bilayer that separates the inside of the cell from the environment is made up of sheets of phospholipids, with a phosphate head that is hydrophilic pointed outwards, and the lipid tails that are hydrophobic facing towards the inside of the membrane. Since these membranes degrade when cells die, this technique picks up the presence of viable organisms, although changes in populations may also be noted because the chemistry of membranes, even within a certain species, can sometimes change when their populations become stressed or are in decline. This flow chart shows how a test for PLFA might be done. After collection and extraction of biotic materials, they are analyzed for their components by gas chromatography for their makeup. The final result is an assessment of the community composition that is often reflected in a chart like the one on the lower right. It gives an idea of who is there and tells us something about functional diversity when the roles of the different groups of organisms are known. There's still a lot to be studied about what these community compositions say about conditions in the field, but they are starting to give insight on how environment and management practices affect what is happening underground. PLFA is an example of what are called biomarkers, a way of differentiating communities by cellular components within them. A focus strictly on activity is another way of understanding what's happening within the soil biome. Microbial activity is often assessed through the study of enzymatic activity within the soil. Most soil functions performed by microbes are carried out through the excretion of enzymes, which help them to break down and digest materials within the system. This chart shows some of the principal enzymes that are looked for and what functions they perform in the soil. The first, beta-glucosidase, is important in the breakdown of carbon compounds and is commonly used in assessment. At the bottom of the chart, urease will indicate production of ammonium, while the last two enzymes help with the bioavailability of phosphorus and sulfur. The chart to the upper right here is another way of representing the breakdown of enzymatic groups by the substrates of carbon, nitrogen, sulfur, and phosphorus, and shows again that the various enzymes listed have been implicated in the breakdown of each. The two bar graphs record the activity of two fundamental enzymes, beta-glucosidase and urease, and not surprisingly reflect the fact that activity of those two enzymes increases in production systems that foster the soil biota. It's interesting to note on the right in the urease chart that the addition of nitrogen, represented by the red bar, suppressed urease activity. This is a phenomenon that has been noted in many situations, specifically that many nitrogen fixation and cycling functions are depressed when supplemental nitrogen fertilizer is added to the system from the outside. Through signaling and intercellular communication, microbes seem to have a way of knowing what is needed and for reacting to various situations. This graph from the Soil Biology Primer of the NRCS merely reflects the fact that microbial activity will increase in the warmer months when conditions are more favorable to microbial community growth. Another measure of activity that has gained a lot of traction in recent years is soil respiration. Microbes are living organisms, and when aerobic microbes make use of carbon substrates, they respire carbon dioxide. The more they are active, the more carbon dioxide they respire, and this is an indication that they are consuming more food. As they consume and digest organic materials within the soil, they release nutrients previously bound up within those materials. Thus, the connection is made between respiration and the mineralization of nutrients. This relationship has been extensively studied and has some implications for agriculture, as we will see. As would be expected, most of the food that is worked through is coming from labile, more easily digested food sources. And the activity is fostered by the right environmental conditions, especially moisture. A common way of conducting the respiration test is to take a dry soil and re-wetting it. This creates what is called a burst of activity or respiration. A real-world example of carbon burst that one can notice is the greening of a pasture after a spring rain. If a field with grass has been relatively dry and then receives some moisture, it is sometimes possible to notice a deepening of green color in the grass, which presumably comes from an injection of mineralized nutrient into the system, made possible by the microbial activity. Once the microbes have gone through the burst phase, activity will slow to more of a status quo level, and this leveling off of activity is called basal, as in baseline respiration. People around the country are starting to use respiration to assess the release of nutrients into the soil from the natural environment, although more work needs to be done in correlating respiration levels to actual releases of nutrients. Part of the reason for this is that testing procedures, both in the field and in the laboratory, can vary in terms of how the soil is prepared, levels of moisture, environmental conditions, and this can give different results that make it hard to make generalizations. Nonetheless, there's a definite relationship between soil activity characterized by respiration and nutrient release. The next few slides were created by Dr. Alan Franzlubers of North Carolina State University, who is one of the leading researchers in the realm of respiration. This graph shows respiration activity of a soil at 3 different depths over an incubation of 28 days. As we've noted several times, the bulk of the biota is in the top 6 inches of most soils, and this is reflected here by the red line showing the upper 10 centimeters of this soil having the most vigorous respiration. All 3 curves show a burst or flush of activity within the first few days, and then level off to basal respiration, which involves a slower release of nutrients. This second chart shows a very clear relationship between soil biological activity expressed through respiration of carbon dioxide to the mineralization of nitrogen subsequently released into the soil. Increasing biological activity is directly related to the release of nitrogen. This increased microbial activity has an implication for nutrient management. What this study shows is that as soil biological activity increases, and therefore mineralization of nitrogen increases within the system, there is less effect on overall yield of a crop with increasing doses of nitrogen fertilizer. This is because in conditions of high biological activity, the microbes are helping to make adequate nitrogen available to the crop, thus making supplemental nitrogen less effective in increasing crop yield. This final schematic summarizes what has been learned about the relationship of respiration to farm management and environmental quality. When biological activity is low, on the left here, presumably there is less carbon and nitrogen containing food within the system, and there is more benefit brought to the system by adding fertilizers. To the right, in sites where there is plenty of available nitrogen and food, the soil biological activity is high, and therefore it does not make as much economic nor environmental sense to add as much supplemental nitrogen, because it would be much less effective in increasing yield and runs the risk of being lost into the environment. The overall goal here is to find a sweet spot of improving soil biological activity through management practices and a nutritional program that adds only enough nutrient to complement nutrients already available within the soil system. The Solvita test is one of the most commonly used field tests for assessing respiration. It is based upon a gel technology that responds to levels of carbon dioxide by changing color. The gels covering the two paddles in the soil here have reacted differently to the higher and lower levels of carbon dioxide, and their respective colors can be compared either visually to a color chart, similarly to how one might look at a pH test, or by use of an automatic color reader. This is a diagnosis of what those colors reflect in terms of soil biological activity, an estimation of the amount of carbon dioxide respired at each level of activity, and what that activity might mean in terms of the release of nitrogen. It's an actual quantification of how much nutrient you're getting based on the respiration recorded. Will Brinton, who founded Woods End Laboratory, which created the Solvita system, created this chart. It's a good reminder of something that we often forget, that carbon is itself a nutrient for plants. There's a huge benefit that comes from respiration of soil microbes, not only in that they are helping to release nutrients in the soil, but by the very fact that they are pulsing carbon dioxide out of the soil right to where the plant leaves are growing. It's well established that plants grow better in a carbon dioxide-rich environment, and this chart gives an estimation of how much carbon dioxide two very common crops take in per acre per day. What it also shows is that the amount of carbon nutrient needed by those two plants can be supplemented by a healthy soil microbiota. Last but not least, I would like to touch upon how the revolution in genetics is contributing to how we understand the microbiome. In the past, scientists had to approach understanding the organisms in the soil one at a time, by observation, by culturing them, by looking at their effect on substrates and trying to identify them individually. Now we can look at them at the community level. Soil microbes are very small, of course, and therefore contain a relatively tiny amount of genetic material. But with amplification techniques that were developed in the late 20th century, it became possible to replicate that material into amounts that could be studied. This led to, and is still important in identifying genes within organisms and understanding their function. This is what is meant by partial analysis, which looks for certain traits. In the past 2 decades, however, a new technique has been developed that allows one to look at an entire sample of all of the organisms within the systems at once. All the genetic material of the entire biota of a soil sample is combined into an aggregate sample, and through computer technology, one is able to assess the entire sum of the genetic material of that sample, which could contain over 10,000 genes. This composite analysis results in a kind of portfolio that shows the diversity of organisms that exist within the system. Thus, it can give a good read on the diversity of the types of organisms that are present, even if each one individual organism has not been identified, and they usually have not been. Where certain genes have been linked to traits, we can get an indication of the functional diversity or the activities that are being performed within the system. This correlation is being improved daily through the cataloging and the inventory of different genes into libraries, both public and private, that are characterizing the genetic material of a wide range of organisms. As the technology advances, the cost is dropping very fast, sometimes by orders of magnitude in just a few years, making it much more accessible. Metagenomic tests can be run very rapidly and are therefore helpful in assessing short-term changes within a system, whether it is in a soil, in the canopy, or even within someone's stomach. The technology is therefore now finding useful applications in agriculture. This flowchart represents how a metagenome might be identified by extracting the DNA from a soil community, amplifying or duplicating that material if necessary, sequencing the genes to identify them, and then taking that composite analysis and grouping the discoveries found to categorize the genome in a way that would be useful in comparing it with other genomes. This is an example of what such a characterization or comparison looks like. It was produced by a company that is performing metagenomic analyses for agriculture. These two pie charts represent large groupings of microorganisms in two samples, one compost and one soil. From the community makeup identified, they are able to tell something about soil activity and to make recommendations for use in the field. Of course, it's not yet understood how all of these groups of organisms correlate to soil function, and we have much more to learn. But as more and more trials are conducted, we are starting to have a much clearer picture on how soil community makeup affects what happens in agriculture. These are a few examples of how metagenomic understanding of community composition and microbial activity can currently be applied to agriculture. One of the most common ways that is used is to help identify particular pathogens or pests within a field. This obviously is helpful to know in advance of planting a particular vegetable crop, for example, that might be susceptible to one of the pests found. By knowing what organisms are in the soil, we can also learn something about how nutrients like nitrogen or phosphorus are cycling or being added into the system. We could also tell something about soil-water relations and whether a soil has drainage problems or is perhaps being overwatered, by whether aerobic or anaerobic microbes are predominating. Understanding all of these interactions and metabolic pathways are increasingly helping us to manipulate and manage the soil biome, and thus helping us to better manage our agricultural systems. I'd like to thank you for joining this series and hope that what we've shared about the soil biome and how it's currently being assessed will help you in the future management of your own agricultural systems. We wish you all the best.